I believe actually in this more or less for the same audience, but quite a few years ago, I've presented something which I will go through briefly again in the beginning of my talk about actomyosin. So this is really the part of the cell cytoskeleton on which I have been focusing for the last few years. But the parts I haven't presented at all, because that was just beginning by then, was about tissue morphogenesis, which will be the heart of the talk today. So, okay, so this is the outline. I'll actually bring you through very different scales. We will start with just a very simple hand-waving model of what actomyosin is. I'll introduce these players in the next slide, and get some constitutive relation for that living material. Then we will go quite up in scale, because this is nearly 0.5 millimeter. So this is an embryo of the fruit fly Drosophila, on which we will have a look at tissue scale flow using concepts from that scale here, using the Regical law there. And then, because there is some missing gap between this scale and that scale, we will go back at an intermediate scale, the scale of a few cells for neighbor exchanges in tissues. So, okay, actomyosin, that's actually two guys. Actin. So actin forms long filaments, which are, so this is a polymer. They form long filaments in the cell. You see them here in a sort of top view of a cell which is on a flat substrate. So you see filaments going in all directions, but with some organization though. And if you do a cut of this cell in the XZ plane between these two lines, you see that actin is really mostly at the periphery of the cell, which is due to the fact that actin is binding with molecules which are actually transmembrane molecules. So actin is attracted and preferentially polymerized close to the periphery of the cell, making it really what we call a cortex of actin around the bulk of the cell. So this is for a cell which is actually in vitro and so cultured on a flat substrate. In vivo, most of the cells, well, many cells are in epithelia, which are layers of cells neighboring one another. Here is a top view of such a tissue. And we will fly through, so going from the middle of the cells to the top of the cells, to the outer part of the cell. So you see really actin, which is fluorescent in green here, is really at the cell edges and forms a dome at the top. This is what you can see also here in this cut of this image here. So you have actin at the junctions between cells here, and actin also at the top of the cell. Myosin now is the other player in this combo actomyosin. This forms much shorter filaments which associate with actin, and I'll come back to this association and the way it works in a minute. But if you see it at the cell scale, so you see if you see an accumulation of myosin in one cell, people have been doing a number of mechanical experiments on that to probe the role of myosin. So, and I'll review some. We've reviewed them with a guy in this paper. So, for instance, if there is a large concentration of myosin in one cell here, and people perform a cut in the tissue in the form of a circle around that cell, so that can be performed with a laser in the tissue. What has been noted for some systems is that you can have a complete collapse of the surface area of the corresponding cell, where really myosin contracts it very strongly. So that's just the surface area. Of course, the cell has a different shape in 3D, but that's where the myosin and the actin are located. Another experiment, it's B and C here. in the same tissue, sometimes early on, you have a weak connection of the cytoskeleton of a cell with the neighboring cells. So this is the dotted blue lines, the very tiny dotted blue lines at the periphery here. If myosin is very active in that cell, then it will contract the cytoskeleton of the cell without changing its shape. Whereas later on in the same tissue, you have much stronger adhesions between the cytoskeleton, so the actin of this cell, and the neighbors. And this time, the effect of myosin is to contract the whole cell, pulling the neighbors around it. Another observation is that if you have several myosin spots in a row, you will have a deformation which is preferentially in the direction orthogonal to that row of myosin. Yet another experiment is to make a cut within the actin of a cell next to some myosin, and you see then that the myosin is pulling and going away from the location of the cut, which is something you're beginning to feel is natural if myosin indeed is pulling on this network of actin. And finally, here in conditions of an anisotropic boundary condition, so you have a tissue which is under tension in one direction, along the x direction here, and myosin is active, it turns out that it also organizes within the cell and gets anisotropic within the cell due to the cue of this force. And if you remove the effect of this force by, again, cutting the tissue around the cell, you don't get this anisotropic organization. So these are some observations, so that this is useful to get an idea of what myosin is doing in this system. But that's also a good way to see what biologists can do and what they can't do in order to probe that system. So you see these experiments are quite different from what an engineer would do to probe a material. But very often that's or ways of probing a biological sample are limited to such interventions, especially in embryos. That's because these embryos are surrounded by a membrane, and usually if you disrupt that membrane, so if you try to access physically the cells below it, you disrupt the processes you're looking at, or you even explode the Drosophila in my case. So with that, I'll start with the first part of the talk, which is which is joint work with the team of Atefas in MSc and his then PhD students, and where we've used an experiment which was very biologically minded of his to try to understand how actomyosin works in terms of its constitutive relation. Okay, so now I've told you actin was forming a network of filaments which are cross-linked. In the simplest model for such a network would be an elastic solid. Of course, you have this meshwork which whose stress is proportional to the strain applied to it. And the way you can obtain this constitutive relation, you can monitor which is called a strand between two crosslinks, say that it will be finely deformed by the gradient of velocities if you're pulling on it. And you have a probability density function of these strands orientation, so it evolves in this way, and here you'll have the gradient of velocity coming in. If you say that there is an entropic elasticity linked with the elongation of these strands here, normalized by some length, then you'll obtain this simple stress-strain relation. Now, what happens if these crosslinks, as is the case in living systems, are not permanent but only transient links? This is something that has been considered a long time ago for rubber-like materials. In that case, a simple hand-wavy way, not the way of Yamamoto, but the way explained actually in Larsen, of handling this this turnover of the crosslinks, is to say that they are being removed at some rate and reattached, but they are reattached once the filaments have relaxed. So they're reattached with R being now 0. So that's this delta function here. And Q just ensures that you have mass conservation. Well, that the total number of strands is conserved. So you imagine that these crosslinks are being detached every tau alpha time and reattached instantaneously, but in a relaxed configuration. And you get a viscoelastic liquid, actually, because, of course, all stresses will relax at a rate which is close to tau alpha. So actually you form a viscosity, which is the product of tau alpha and the instantaneous elasticity of your network here. And you obtain Maxwell equation with the upper convected time derivative here of the tensor. Now we have an additional thing because our network is additionally consuming energy via these active crosslinks, which are myosin. So what they do is that they actually perform some walk along the filaments. So one way to really, again, in a very hand-wavy way to model this is to say that they perform some diffusion along the filaments. And of course, they will shorten the filament at the bottom here and lengthen the filament at the top. This appears like something like that. You remove some filaments of some length, you add some filaments of the two other length. This would seem to be something that should average out and give 0 in my equations if I don't dissymmetrize things further, because I have lengthened one filament and shortened another one. It turns out that there is a remaining term which is proportional to the square of the distance that the myosin head travels in one step. Well, that's because of the growth, which is to consider for the filaments is the geometric growth and not just the added length. So it's a product, and it turns out that you have an L squared coming out. There may be other ways actually to obtain a similar term without an L squared by dissymmetrizing by hand things. So there are good reasons for that molecularly. But in the end, you always end up with a term which is a positive term that you add here to this constitutive equation, meaning that you have an added stress, which comes from this myosin effect, and which will be oriented along the filaments that you have. So if you have a preferred orientation in this ensemble average of orientations, then it will be along that orientation that you have this active term coming from myosin. Okay, so I'll briefly discuss what this constitutive equation gives in terms of behavior. This was the subject of my seminar 6 or 7 years ago with you, and then I'll go up in scale. And of course, don't hesitate to ask questions in the course of the talk. So the first thing I'd like to do is briefly, without going into the details of that, of course, compare that with the active gel model, which has been developed around Frank Jüdischer, Jean-François Joanny, and Jacques Pro in ESPCI and Institut Curie, and which is based actually on their work, the previous work on liquid crystals. So instead of having this vector R that I had, and which elongates with the flow and is directly linked with the stress tensor through entropic elasticity, they use a field of polarity which has always a modulus of 1 based on liquid crystals. That actually accounts for the fact that they have here co-rotational derivative rather than the upper convected derivative. And they have a conjugate field, which comes from Frank energy for liquid crystals coming in. Otherwise, the models are quite similar, actually. They have an active term, which is here, which is similar to mine, except for some little details linked with their P field, which is different from a R field, slightly. But globally, once they do their usual simplifications, this yields models which are quite similar, except mostly for the choice of the objective derivative here. So in my model, so I've added here the effect of a solvent that you would often have in a model of polymeric liquids. Actually, I'll remove that and I'll explain you why very soon, and rather use a second viscosity or bulk viscosity here to prevent the divergence of V from being too large, and also because that's a usual term, although it doesn't come out of the derivation that I've shown, and that's usual also for this sort of derivations. I'll explain to you why I believe you shouldn't have incompressibility. So this is a cell viewed from the top when it has just been spreading to a circular shape which is imposed to it. And you see that, so it's in the course of a few hours, and you see that really the flow that you're seeing of actin is really not divergence free. You really have an inward flow, especially of the orthoradial filaments here, which is very strong and endures for a very long time. Actually, those filaments which are radial also undergo the same flow. This can be shown, but they are being polymerized at their outward end, which compensates for their presence. And that's the same for the orthoradial filaments, which are being created de novo here. So if you consider mass conservation, actually, what this means is that saying that you are in the permanent regime and d rho dt is equal to 0 doesn't lead to divergence of V equals 0 as usual in fluid dynamics. It is rather that because you have a regulation of some sort, so new filaments being polymerized in places and depolymerized in others, which actually leads you to the fact that rho is approximately a constant, but it's regulated by the biology, the metabolism of the cell in some way. So I'll just take it effectively. I consider that the density is maintained constant, but not by the fact that you have a divergence-free velocity, rather by the fact that you have a reaction term that completely dominates the mass balance here. Okay, so in that case, so we can consider the a 1D version of this model and put it in simple binary conditions just to see what it gives. So if you have Dirichlet boundary conditions and you prevent any contraction of the material, what you get is after a transient, possibly for stress, you have the stress equal to the myosin imposed stress. So that means if you have a sample of actomyosin between two boundaries, you're feeling, and you maintain these boundaries, you're feeling the stress that myosin exerts on the boundaries, which is what we expected. And it's an inward stress. So you really have to pull outwards to maintain the boundaries. If on the other hand, you let go and there is no stress at the boundaries, then you'll have a rate of strain, which is negative and constant. That means that your sample, because it has no resistance to volume changes here, and it's 1D, you'll have a complete collapse of this network, an exponential collapse of it, with a rate which is again set by the myosin activity here. And because of this turnover of the actin filaments that I was showing you, this is not completely incompatible with reality, at least in the first order. Okay, so then of course it's more interesting to have more complicated boundary conditions, and that was the topic of our work with Etienne. We had a spring-like setup to put in series with the cell, and mostly it's actomyosin. And so we could show the properties of this, and what is interesting is that, so because it collapses to 0 length if the external stiffness is low, and it, on the other hand, reaches a constant force if the external stiffness is large, this stiffness here, you have something like an emergent spring, actually, something which has a set length and set internal stiffness. And actually, well, of course, this is not realistic for cells that they will contract to H equals 0. But if you combine that with the fact that you're polymerizing new filaments at the periphery, you reach really the behavior of something which is like an emergent spring in series with the external spring here. So that was the topic of this paper with Etienne. And so I won't comment on that much more for today, but I'm happy to take questions on that if you have. So now I'll switch to the second part and go up in scale. Unless there are questions, maybe that's the right time for that. Okay, okay, so I'll carry on with this much larger scale. And this time, this is a collaboration with two groups of people. So one is the group of developmental biologists in Cambridge, led by Bénédicte Sanson, who've actually justified the interest for this work. And the other is with Mahamar Diko, who was a PhD student co-supervised by Pierre Saramito and myself. So in the Applied Math Laboratory in Grenoble. Okay, so now we have many cells. That's the surface of Drosophila, which is an ellipsoidal shaped embryo with cells only at the surface. So it's a thin viscoelastic layer of cells, which is undergoing very large movements during its morphogenesis. So I'll play that again. You see that these cells jiggle around a little, and then there is a strong flow towards the ventral side here, and then posterior and around the posterior here. And we'll be mostly looking at what happens from the ventral side here, where most of where you have the largest strain rates actually in this flow. So one great thing with this team in Cambridge is that they're able to track the movements of these cells in 3D in the course of time and really see where one individual cell is going and how it deforms in the course of this flow. And so this allows to have maps of the velocities, and this is very interesting to do some mechanics on this tissue. And you see actually it's 3D, so they can image with the laser sheet microscopy. That's just for really the cheek of the Drosophila, so just this part here. But you see the curvature of that and how this is transformed into cells of which we have the motion. So in order to perform this motion, so that there is very few cells added or removed, nearly no divisions and no cells disappearing into the bulk of the embryo. One very important mechanism is called intercalation. So if you watch these two yellow cells, I'll play that again, they start off not being in contact and then gradually are brought into contact. So there are many which are doing so. This is called cell intercalation. Watch the two yellow ones here. So you see that they're brought in contact. So that means that you actually reorganize the microstructure of this tissue, and that allows the tissue to elongate in one direction and narrow in the other without having too much of cell shape change, actually. Although you see that they go from quite elongated in the vertical direction, which is called DV, dorsoventral, and they are a little more elongated along AP here, anteroposterior X, after a while. So having these flows of, well, these velocity maps, what they could do is calculate the strain rate of the tissue in every place. So this is a strain rate based on the movement of the centroids of all those cells and color-coded for positive and negative strain rates. So you see that really it elongates along the horizontal direction here and narrows around the vertical direction. But actually, because you have these intercalations, of course, this strain rate is relevant at the tissue scale, but doesn't tell you what cells by themselves experience. So you can calculate another strain rate, which is the one of individual cells. One cell can be fitted by an ellipse, and you have a look at the evolution of this ellipse in the course of time, and that gives you a different strain rate because the difference between the two will be the intercalation. So that offers a continuous measure of intercalation. So it would be, and that's one of our projects, extremely interesting to try to give really a rheology that that delineates these three components, and we are progressing towards that. For today, for this second part, I'll focus on having a look at whether I can predict this type of flow on a global scale. So taking a continuum model, forgetting the fact that this is divided into cells, and trying to see whether my continuum model can reproduce the global flow.
Sorry,
So,
Jocelyn, can I ask
sure.
a quick question? Sorry if I missed it, but why is the motion 2D planar? Is it something, is it like that, or is it?
So it's really, so this embryo is really, I should have shown actually a cut of the embryo too. So that's really a single layer of cells surrounding what is called the yolk, which is a huge cell of just serving as food. But this is not a tissue, and there is not much. It's mostly a viscous fluid inside. And really, the dense layer of actin is really at the periphery of this embryo.
Okay.
So in the bulk of it, of course, there are difficulties to observe because it's not completely transparent. We now we know that there is a little more than what we thought, but the most of the mechanical well, the structures, the most important mechanical structures are really at the periphery.
Okay. Thank you.
There's very little extracellular matrix, for instance, at the down below at the basal side of cells. Okay. So yes, so indeed we have an approach, and that was the standard approach for years, of really looking at it as a surface flow. And this surface flow now is driven by myosin, as you would have expected, given the introduction. And actually, in this region of the embryo, probably you could guess here that the myosin is mostly along the edges of the junctions of cells, which are shown vertical here, so DV orientation, and there is much less myosin along the AP horizontal direction. So we do have what I was suggesting before, a preferred orientation of the filaments on which myosin is acting, and we will have an active stress tensor, sigma A, which is preferentially along a specific direction, which is the DV direction, so along here, along the small axis of this embryo, in all places which are green here, which are non-zero sigma A. So sigma A is also multiplied by a constant, which I forgot here, but not a constant, but a scalar function, which is space dependent. And then it has this orientation all along. So okay, so the effect of the myosin is directed here. The rest of the model is just the surface version, curved surface version of what I was showing you in the first part. And I've added here friction because there is this membrane which is just at the outward surface of the embryo, with which there is probably some mechanical interaction, but not nothing active, just a passive friction, viscous friction here. Okay, so this is the model that we've taken to, and which is really the model of the microstructure that I've just postulated that maybe it does work at the tissue scale, and we'll have a look at what it gives. So actually making that work on a curved surface, so this is a compressible flow. You can't solve it by taking a stream function, as many people have done to solve flows on curved surfaces. So this has made a good applied math PhD subject for Mahamar, who's been working on that and has developed this method. So in the end, you can measure the surface here and solve this surface flow on it with the data of this distribution of myosin in the green region and this orientation, and you do get a flow which is what is observed in Drosophila. So this is here this in these green regions, you have myosin, which is acting and which is really pulling the material, and I'll explain that just a little more in a second. You can especially study the effect of the key parameters. So one is the friction, the hydrodynamic length, which results from the balance between viscous friction and the viscosity of this fluid at the surface. So if you have a very large hydrodynamic length, you have just one large vortex on the whole of the embryo. And as you decrease it, you have a flow which is more and more local in the region of a strong gradient of myosin. So that's really where you have the divergence of the active stress, which is non-zero. So that's really driving the flow. Then you can study also the effect of the bulk viscosity. So this is what is making really this flow work, because of course, so I've told you the myosin was contracting the material in this green region. So if you have no resistance to variations of area on the surface, what happens is that the whole of the material flows towards this ventral side, but can compress, and there is nothing to prevent it from contracting along this dorsal ventral direction, and nothing to promote the flow towards the posterior here. Whereas if you have a second viscosity, then there is some cost of doing that, and you'll have a flow which is going towards the posterior of the embryo, because you have a cost for varying the area of the cells here. So in the end, exploring the range of parameters, and without even doing fitting, we could find a range of parameters for which the flow is really close to what is observed in the embryo. We've had to tune a little our active stress pattern to match the features of the actual embryo, but we have a very good agreement in terms of the flow, and we can even compare the strain rate. So even in the a mathematician would say that we have an H1 convergence to the to some good measure of what is happening in the embryo. So the strain rates compared along this line here and that line here are actually quite close to what can be measured in vivo, which is the very spread around dots, and the mean is the black line. Okay, so we do have some agreement, and with this very simple model for this global flow with cells, although we've had to choose second viscosity eta b, which is quite large and much larger than what I would have expected, because we have no mechanism really to explain at a molecular scale why this actin meshwork, which could thicken in the third dimension, for instance, or could densify much more than what we see, would resist that much area variations compared to resisting shear. So that's why I was telling you this model really also, this work, the good results of these works also needs going back to the intermediate scale to link between our molecular scale model of actomyosin and what is really happening at the tissue scale. And that's my third part, if I'm not too late. That's it. Yes. Okay, so this one is still in collaboration with the group in Cambridge, but in addition, a young researcher, a mathematician, Alexander Nestor Bergman, has joined it. He's done his PhD about Vertex models, which I'll tell you about in a minute. And Alexander Fletcher is also a specialist of Vertex models, who's been working on tissues for quite a long time in Sheffield. So we go back to this cell scale and have a look at this intercalation process, which is indeed driven by actomyosin. So actomyosin is here, contracting this edge, and this leads to this rearrangement of cells. So the way this has been tackled by very many groups in the past is the so-called Vertex model, which has the advantage of being extremely simple and fast. It is simply saying that each cell in this tissue has a target area, missing a square here, and has some line tension of its perimeter, and you minimize this with just something which is rather a pseudo time, because here this friction is the friction associated with vertices, so it doesn't have a real link with the physics of the problem. So it's really a way of minimizing this energy functional by rearrangements. And then here in this video, you see the cells which are highlighted in color are those which are connected with a vertex with more than 3 cells surrounding it. So a vertex which is undergoing a change of neighbors. And you have to do that actually manually. You have to change the topology manually of your mesh when this happens. So this is very simple and convenient and reproduces a lot of the effects seen in vivo, but you don't get much from it in terms of understanding what really underlies it in terms of the link of the microstructure on the global behavior. So we wanted to extend it, but that's very difficult actually to find what is the relevant degree of freedom one should add to this model. And it's also, we are trapped in a minimum of simplicity, in a maximum of simplicity rather with this model, and it's difficult to get out of it. So we actually went as far back as modeling each cell as with its cortex, so the actomyosin at its periphery, as a viscoelastic rod in the sense of a Kirchhoff rod really for its elastic regime. So they are subject to a stretch and a bend elasticity. They have a reference configuration here and zip up with neighboring cells, neighboring cortices through adhesions, which we've modeled as a set of springs. So we have springs everywhere there's space on these cortices, which maintain the cortices close together here. And actually, we can see in super resolution microscopy here that this is not completely unrealistic. You see that these tricellular vertices here are actually have a shape a little like this. So you do have this sort of configuration in vivo. So the way we actually now the way we implement active mechanics, so myosin effect on that, is to have to use morphoelasticity. You have a virtual configuration in which you say that the equilibrium length of some portion of this cortex is actually shorter because myosin has been working on it and contracting it. So this is a pre-strain approach rather than pre-stress this time. And then the elastic, mechanical balance is calculated from this virtual configuration to the current configuration. So this is the algorithm that is used here. So there is one part about the adhesions, setting the adhesions with these springs, then applying the pre-stretch, and finally solving the elastic equilibrium for these cortices in every place. And to show you, well, that there was quite some developments in order to do that from the energy formulation. But that does work, and are surprised with that. It's so nicely behaved, actually, and you can perform these simulations without too much difficulties, given the rather large complexity of that. Then I've told you it was viscoelastic rather than elastic. So what we do is the poor man's viscoelasticity in the first place. So at every time step, so every characteristic time tau cortex, tau alpha here, we actually completely relax the elastic stresses by resetting the stretch, the current stretch between the reference and the current configuration to 1. That corresponds to saying that elastic stresses have completely relaxed over this length of time. And actually, you can just do a little hand-waving argument that this does recapitulate what happens with the Maxwell model for the stretch of these rods. So if you relax, if you say that the reference configuration obeys a relaxation equation like this, you actually find that for small lambdas, for lambda close to 1, you have something like a Maxwell equation with a pre-stress here. And this pre-stress is this is related to the pre-strain that you've imposed between reference and virtual configuration. Okay, so now with this algorithm, you can put cells together and let them interact with a boundary and other cells through adhesions. And quite naturally, they zip up together as you increase adhesion or decrease bending stiffness, or do both. So this is here the size of this opening here at the tricellular vertex as a function of these parameters. And now you can simulate neighbor exchanges by making this junction contract. Then at some point you reach a four-way vertex, what we call a four-way vertex, and there is no contraction anymore. But adhesions start appearing between the two other cells, which were not touching one another before, and this junction here extends. So this extension is purely passive. This is because in any vertex model, if you have the same, in any such configuration, if you have the same tension in all edges, of course, the 120 degrees angle between different line tensions is the only one which is at equilibrium. So you can extend passively something that was originally at 90 degrees angles with 4 cells. So this does work and recapitulates the in vivo behavior, which is quite fine. But there is, so the big player here, the key player, is actually the rate at which you have an evolution of these adhesion molecules. So remember, we have small springs between two morphoelastic rods, which are free to shear actually and to move relative to one another. If you have a very fast turnover of these springs, what they do is mostly maintain these cortices in contact, but they don't add much cost of shearing these two vertices. As a result, what happens in the simulations, which is not the one I've shown you, is that the tension that you generate here in this shrinking edge is mostly balanced by tension all around the perimeter of that cell. And there is not too much which is transmitted to the neighboring cells and to the boundary conditions of your tissue here. And you have mostly the same, you have slightly lower tension in the cortex next to the cell which is contracting. Whereas if you have a slow turnover of these adhesion molecules, then you'll be able to really bend them and also play in the tangential direction with these springs. You see here actually around this vertex that they are bent and disorganized. Not disorganized, but actually, there is elastic stretch here. And you transmit actually the stress which is originating from this cell 1 to the neighboring cells and towards the boundary condition. So you have something which is much more a global balance of the tissue. And that changes dramatically actually the dynamics. If you compare now the two conditions, so fast adhesions here and slow adhesions there, here you see that it's mostly those cells which actually shrink away, their perimeter disappears, and actually the material from the cortex, the rest of the cortex of cell 1, for instance, flows into the shrinking junction and is being shrunk by myosin until this junction completely disappears. But in the course of that, of course, if you shrink the perimeter of that cell too much without putting much on the others, what you have is a strong variation of the area of that cell, because we have no area term at all in this model. Whereas if you have a transmission of the stress towards the boundary conditions, which are Dirichlet far away there, then you do have a different balance, and you shrink much less those cells, and they preserve their area much more. Actually, this is the curve of area evolution in the course of a neighbor exchange here, where this is the length of that junction in negative and the length of the new junction in positive here. And if you have slow adhesions, this is much more like what we have in vivo, where we have a small variation of the area of cells which are undergoing neighbor exchange, but which is much closer to what we have with slow adhesions here, slow turnover of adhesions. Okay, so maybe I've been a little, no, I haven't been too long actually. But so that's what we could say with this model. We still have to validate it experimentally, because we predict these strong flows. I'm missing some picture here. We predict these quite strong shear flows, or the importance of these shear flows between the two opposed junctions, which hasn't been looked at experimentally yet. So we want to do that with Benedict in Cambridge, and need to quantify it. But then the next steps is really to continue bridging this the molecular and cellular scale towards the tissue scale, and try to have a constitutive relation which takes into account the behavior that we're describing here for the tissue scale. And especially if we can work out how this area preservation may work with based on this behavior. So we have two ideas for that. That there could be kind of brute force micro approaches based on simulations, or we could also have now go back to the Vertex model, and now that we know which key new degrees of freedom we need to add, really these flows across vertices and the cost of shearing between two opposed cells, to obtain something which would take into account these effects in a simplified model. Then another perspective is well, that's in line with the questions, the question you've asked already, going 3D, because of course, so this is just a model of a surface. We know that most of the interesting things happen on that surface, but the rest of the tissue deeper down is known to have some effect on these dynamics, and we're beginning to have quantitative data on that from biologists. So we can begin to address that, but there are many questions on that actually, and that's a whole other talk, I'm sure. So with this, I think I'll conclude here, and I'm open to questions.